Hello guys, I hope you guys are doing okay. I am Mambe, your favorite life sciences teacher. Thank you for tuning in. Please subscribe, like, share and comment. Before we get into today's lesson, I want to apologize and make a disclaimer about the last video that I've uploaded, which regards to which focuses on chromosomes and crossing over. So I said the last two pairs of the karyotype represent sex chromosomes. It's not the last two pairs, guys. It's the last pair that has two chromosomes, which can be XX or XY. That is the only pair that has or that represent sex chromosomes, which are gonosomes. I'm sorry about that mistake. I couldn't pick it up when I was editing, but yeah. And then today we are going to learn about meiosis. We're going to learn about meiosis 1, meiosis 2, and also the diagrams that are involved during these processes. Anyway, so the question should be, what is meiosis? Why is it important? And why are we studying it? So meiosis is a cell division process whereby one diploid cell divides to form 4 genetically different daughter cells. And the daughter cells are actually haploid. The first cell is diploid, it's a somatic cell, but at the end of the day, it's going to divide to make 4 haploid, genetically different sex cells. So that is meiosis. Now, unlike mitosis, we've learned about mitosis from grade 10 or the previous classes. Unlike mitosis, meiosis has 2 cell divisions. So we have meiosis 1, we also have meiosis 2. Now, in meiosis 1, we're gonna start with the phase that happens before meiosis takes place, and that is interphase. So I need you to know this. I want you to know these letters. So it's IPMAT or IPMET, depending on how you pronounce it. You know the I represent interphase, prophase, metaphase, anaphase, and telophase. That way you'll be able to know which ones follow the other ones. So during interphase, this is the process whereby DNA replication takes place. We spoke about that. And then we are going to have prophase, which is the stage whereby crossing over takes place. Anyway, so all those things that is happening, remember we said one cell divides. So it tells us that we are actually dealing with a cell. So this whole thing occurs in a cell. The first outer layer represents a cell membrane. The first outer layer represents a cell membrane. And as always, if you see the spaces, the space represents a cytoplasm. So this is our cytoplasm. This is our cell membrane. We also have a nuclear membrane. The nuclear membrane is the one that has dotted lines, but the one that has a solid line is actually a cell membrane. The one that has dotted lines is nuclear membrane. And then inside the nuclear membrane, we are going to have chromatin networks. I did not write it, but I'll make sure that I include it in this video. So these are our chromatin networks, which are going to make chromatids, and then the chromatids are going to make our chromosomes. So we also have what we call centrosomes. So what happens in your examination, they bring these diagrams and then they'll ask you to actually label them, or they'll give you this diagram and ask you which phase does this diagram represent. So this is our centrosome. Our centrosomes are going to help us to get our spindle fibers. We have our nuclear membrane, we have our chromatids, and then we have our cytoplasm, and then we also have our cell membrane. Now we are going to the second phase, which is the prophase. So you need to do this every time. If this is prophase 1, you need to tell us this is prophase 1. If they put this diagram in your test and they ask you which phase is theirs and you just say prophase, that is a 0. Because you're not telling us which prophase, especially if we are talking about meiosis. If it was mitosis, you were just going to say prophase because it's only one cell division. But in our case here, we have prophase 1, we also have prophase 2. What you're going to do is to tell us this is prophase 1. If it's prophase 2, you're going to tell us this is prophase 2. Now in prophase 1, we still have our cell membrane. We also have our nuclear membrane. Inside our nuclear membrane now, we have what we call chromosomes. And as you have learned from the previous video, these chromosomes are in pair. So it means that they are called homologous chromosomes. Now, from our centrosomes, we are going to have the formation of our spindle fibers because we're going to need it to attach to the chromosomes. So this is prophase. Remember the process that we mentioned, it happens in prophase, is crossing over, meaning that the following step we are not going to see the same chromosomes. So we should see that actually crossing over took place. And the first stage is metaphase. So metaphase is the process whereby these chromosomes are aligning along the equator in homologous pairs. So they align along the equator. You can take the M as in like middle. They align in the middle. So the middle of the cell is called an equator. So this is our, my God. So the middle of our cell is going to be our equator. So the chromosomes, they align along the equator in homologous pairs. If you look at these chromosomes, you can see that this one was clear, but now it has some dotted lines to show that crossing over did take place. You can see the same thing from the other 3 chromosomes. So the crossing over that took place in this process then resulted into these chromosomes. Now, as they align along the equator, they align because we have what we call spindle fibers. So our spindle fibers are attaching to the chromosomes. They are the ones that are attaching the chromosomes, or they are the ones that are pulling the chromosomes towards the middle of the cell. So now we have our cell membrane. In metaphase, our nuclear membrane disappeared. In metaphase 1, we have chromosomes aligning along the equator in homologous pairs, and then we also have the disappearance of the nuclear membrane. So we move into the 4th phase. The 4th phase is anaphase. So what happens in anaphase is that the homologous chromosomes now, they are separated. Remember, we need to have a division. So they are separating. So our chromosomes are separating in anaphase 1. So what makes the chromosomes to separate is again the spindle fibers. So now the spindle fibers are pulling the chromosomes towards the opposite poles. The other chromosomes will go to the left, the other chromosomes will go to the right. So that is what is happening in anaphase. We also have a condition that is called non-disjunction. Non-disjunction is the process whereby the chromosomes fail to separate from each other, or rather the chromatids in anaphase 2, they fail to separate. It might be that this chromosome now is no longer on the other side, but it goes with the other 2 chromosomes. So it's going to cause a disorder in a human being. So that is what we call non-disjunction. Remember this condition. They love it so much. So we still have our cytoplasm, we have our cell membrane, we have our chromosomes pulling away from each other towards the opposite poles. We also have what we call a centrole, and then we have our spindle fibers, okay, and then we have our spindle, and then we have our spindle fibers pulling the chromosomes away from each other. So we also have the last step now. The last step is what we call telophase. So what happens in telophase is that this is our last step. We said the cell is going to divide to form 4 cells, but before it forms 4 cells, the first stage is going to form 2 cells, and the 2 cells are going to further divide to form 4 cells. So in telophase, we have the division of our cell. So now The chromosomes have separated towards the opposite poles. We have the formation of two cells. Now there's a process called cytokinesis. Cytokinesis is the point whereby the cell divides into two. This is the step. And this is the process, but what we call here, the middle part is called cleavage. The middle part is called cleavage. The process that results in cleavage or in two cells is called cytokinesis, but the middle part between the two cells is called cleavage. So now we have our last step, which is telophase. We have to move again to meiosis 2. So with meiosis 2, we are not going to have interphase because already we did have interphase. Already we did have the formation of chromosomes. And then what is going to happen is these two cells are going to further divide. So this cell, even if I make a presentation that shows one cell dividing, it means this cell is going to go through the same process, and then this other cell is going to go through the same process, so that we can end up with two cells here and then two cells here, and then they are going to make four genetically different haploid cells. Without further ado, let's go to the meiosis 2. So the diagrams are now representing meiosis 2. Before we discuss them, I want us to talk about the differences between meiosis 1 and also meiosis 2. So with meiosis 1, there is interphase before meiosis 1. However, there's no interphase in meiosis 2. And another thing is, in prophase, we have crossing over in meiosis 1. In meiosis 2, we do not have crossing over. Chromosomes in meiosis 1, they arrange in homologous pairs. However, chromosomes in metaphase 2, they arrange individually along the equator. The other ones are homologous pairs along the equator. The other ones are individually. And also the number of chromosomes in meiosis 1 is halved, but the number of chromosomes in meiosis 2 is not halved. And also in meiosis 1, we have one cell dividing to form two cells. However, in meiosis 2, we have two cells dividing to form four cells. Those are the differences that you need to know. If they say tabulate, you're going to tabulate and write the differences that I just told you. So you talk about chromosome number, you talk about crossing over, interphase, homologous pairs, and also the division that happens, how many cells actually divide. So these diagrams now are going to show us, or they illustrate the whole process of meiosis 2. So these are our two cells that we got from the first division, the first meiotic division. These are the cells that we got. So we continue with the cells that we had. This is cell number 1, this is cell number 2. As we start on prophase, we already have chromosomes, and then we already have our centrosomes, which are going to form our spindle fibers. So we can already see that this is the formation of spindle fibers. Now, during metaphase, in meiosis 1, the chromosomes were aligned along the equator in homologous pairs. However, in metaphase 2, the chromosomes, they align individually. It's no longer homologous pairs. So they align individually on each cell. So cell number 1, they align. Cell number 2, they also align. So now we move into the other stage, which is anaphase 2. So with anaphase 2, we are going to have, remember in meiosis 1, we had the homologous chromosomes being pulled away from each other. Now they still need to do the pulling. The spindle fiber needs to be pulling something. But since we do not have the homologous chromosomes, it means one chromosome is going to be pulled to the opposite side. And remember, a chromosome is made up of two chromatids. So the other chromatid will go to the left, the other chromatid will go to the right pole. So this is what is happening. The same thing that happens in cell 1 is going to happen in cell 2. the chromatids now, not the chromosomes, the chromatids are being pulled towards the opposite pole so that we can get our division. The same thing here happens. And then we are going to have the last stage, which is the telophase. With telophase, we already have our 4 cells. This cell divided to form 2 cells, and then this one divided to form 2 cells. So all of them are in their own nucleus. And we already know, if we have this separation, there's a process that took place, and that process is called cytokinesis, whereby the cell divides, and that is basically meiosis 2. So meiosis 2 is actually the simplest. You just need to know that in meiosis 1, we start with the actual chromosomes, or the actual homologous chromosomes, but when we deal with meiosis 2, we actually have individual chromosomes. Another thing is meiosis 2 kind of relates to mitosis, because mitosis deals with single divisions. However, it doesn't result into 4 cells, but it results into 2 cells. So that is it for today's lesson. If you have any question, leave it in the comment. If you want me to cover any topic, just say whatever that you want to say. I'm with you, and I hope you're going to make it in your examination. Thank you. Bye.